Okay, so get started. Welcome back. Hope you guys had a good break. So I've got a couple of videos working to show you the structure of a neuron, and two, how the neuron actually sends its signal to adjoining cells. So we'll listen to that for a start.
Activities and physiological functions.
We'll try that again.
Neurons are the specialized cells that control and monitor body activities and physiological functions. They sense changing conditions, process sensory input, and direct the body's responses. Neurons come in many different shapes and sizes, but for all of them, the cell body, also called the soma, contains the nucleus and most of the other organelles. Extending from the soma are branch projections called dendrites. Their job is to receive information from the extracellular environment, from other neurons, or from other specialized cells. A typical neuron also has a long process called an axon that carries information that will be relayed to another neuron or a different cell type. The axon ends in fine extensions called telodendria, each of which has an expanded synaptic terminal at its tip. A synaptic knob, shown here, is one type of synaptic terminal. Inside each synaptic knob are synaptic vesicles containing chemical neurotransmitters that, when released from the synaptic knob, affect the transmembrane potential of another cell.
Okay, so we covered this information regarding the synaptic vesicles and the synaptic knob, and how the neurotransmitter is then transmitted across that synaptic cleft. That gap that you actually see, it is a physical gap. You can see in this electron micrograph right here. You can see that gap right there. So it is a real gap. So these are chemical synapses. So we do have some electrical synapses in our body, especially in the heart, for example. And those electrical synapses, their membranes do actually touch each other. And their membranes have channels between the presynaptic and the postsynaptic cells, so that the current can actually flow through. Right? But around 95% of the synapses in the human body are chemical synapses. So this is the type of synapse you get.
A neuron relays information to another neuron at a synapse. The neuron transmitting the information is called the presynaptic neuron, and the neuron receiving the information is the postsynaptic neuron. Let's look at what happens at a cholinergic synapse, one of the most common types of synapses. When an action potential arrives at the synaptic knob of the presynaptic neuron, voltage-regulated calcium gates open. Calcium ions enter and bind to synaptic vesicles. This leads to exocytosis, which releases the neurotransmitter acetylcholine from the synaptic vesicles into the synaptic cleft. Acetylcholine molecules then diffuse across the synaptic cleft and bind to ACH receptors in the membrane of the postsynaptic neuron. The binding opens ion channels and the membrane depolarizes. If this depolarization brings the initial segment of the postsynaptic neuron to threshold, it will result in an action potential. The depolarization of the postsynaptic membrane is short-lived because acetylcholinesterase rapidly breaks down the ACH in the synaptic cleft.
Okay, so that is a very rapid, quick introduction to signaling at chemical synapses in the nervous system. So these neurotransmitters then, they are chemical messengers. They are released by the presynaptic membrane. Note that the vesicles themselves don't get released. As I showed you there, the chemicals get released. Okay? So the vesicles fuse with the membrane and then open up, and their contents are released. Those neurotransmitters that are released then bind to receptors on the postsynaptic membrane, causing the receptors to change their shape, which then permits sodium ions, for example, to flow into that postsynaptic neuron, regenerating that entire message all over again. Now, when those neurotransmitters are bound to the receptors, once their message has been transferred, the neurotransmitters are then broken down. Okay? They're broken down by those enzymes that exist in the synaptic cleft. So in this cleft here, you also have enzymes that can break down these neurotransmitters that are released. Because if you don't break those neurotransmitters down, they'll stay bound. And as long as they stay bound, that postsynaptic cell is going to be stimulated. Okay? And if the intent is not to keep stimulating that postsynaptic cell, it could then lead to consequences of overstimulation, which could result in seizures as well. We went through this, correct? Did we speak about the afferent? Where did we stop? Sorry? We haven't gone through this? No? Okay. So neurons can be classified based on whether they carry sensory information, whether they carry motor information, or whether they transmit information between the sensory and the motor system, and therefore they're called interneurons. Interneurons have other functions as well. Okay? So sensory neurons, also referred to as afferent, with an A in front of it, afferent neurons, that should not be brain, that should be bring. Okay? So they bring information into the central nervous system. All right? So sensory information, for example, you touch something, if it's hot, that sensory information is brought into the nervous system via afferent neurons, which are sensory neurons. Those sensory neurons then generally make connections with interneurons. Remember, we mentioned that interneurons are very small. Okay, they're not like a long sensory or long motor neurons. They're very small, but they have significant function in the nervous system. All right. So if you, for example, you have a sensory signal that comes in that informs the nervous system that you need to act. So, for example, you've got a hot pan on the stove, right? You forget that you see the water boiling over. You forget that it's hot. You actually touch the pan. You hold the pan. The metal handles. What would you do? Pull your hand away, right? It's automatic. It's a reflex. Now, if you've got a 2 or a 3-year-old standing right next to you, right, they're about yay high. Now, if you pick that pan up, that intention to actually drop that pan, that reflex is overridden, because you know that if you drop the pan and you splash the water, that toddler is going to get burnt, right? So this is where these interneurons can come in, and they can fire other systems that override that reflex, and they prevent the reflex from actually occurring. They can inhibit those reflexes from occurring. That's just one example of what these interneurons can do. So that sensory information then comes in, and if a motor response is required, they activate the motor neurons, referred to as efferent neurons. Efferent neurons. And these efferent neurons then take information from the central nervous system to the periphery. Okay? So they take information out of the central nervous system to the periphery. So we have two main types of sensory neurons. We have visceral sensory neurons. These are the ones that monitor the internal environment, such as those inside the body, such as the organ, heart rate, for example. All those are visceral structures. And then we have a somatic sensory neuron, which monitors the external environment. Okay, so these are the receptors that are underneath your skin, the sensory receptors, numerous sensory receptors, right? Visual, auditory, touch, etc. Sensory receptors can be divided further into interoreceptors, and that's what they actually do. I'm not going to go into detail because you can read and understand. Exteroceptors. Now note that exteroceptors do not have to be at the surface of your body. Okay? So if you shine a bright light into your, or if you're looking right now, you can see what's up there. Where are the receptors that are picking up that signal? They're in the retina. They're at the back of the eyeball, right? They're in the retina. But the signal that they pick up are external. Therefore, they're classified as exteroceptors. Okay? Same with hearing. The hair cells are in the cochlea, which is deep inside. But the signal that activates those hair cells are from outside. So hair cells as well are exteroceptors. So they do not have to be on the surface in order to be exteroceptors. And then we have proprioceptors that monitor position and movement. And therefore, they're in joints and skeletal muscle, for example. Motor neurons carry the information from the central nervous system in response to the sensory information that comes in from the central nervous system to the peripheral organ or effector, and they're carried by the efferent fibers or motor fibers. So this gives you a bit of insight onto what these interneurons are. They're located mainly in the brain and spinal cord, as well as in autonomic ganglia, autonomic. And they're responsible for distributing that sensory information as well as coordinating the motor information as well. Okay? They're also involved in higher functions such as memory, planning, and learning. So they don't just transmit messages. They process this information. Okay? And they can actually coordinate the response to the sensory information. Now, as I mentioned a couple of weeks ago, around half the volume of the nervous system, about 84 billion cells, are these neuroglia. There are numerous types of neuroglia in the central and peripheral nervous system. Some of them in the nervous system are these astrocytes. You have microglial cells, okay, and other cells as well. So looking at the astrocytes, they're very important in regulating substances that enter the central nervous system from the periphery. So as you can see here, you have a capillary blood vessel, right? And the foot processes of these astrocytes, what they do is they actually form a coating around all these blood vessels. And they regulate substances that can come out of these blood vessels. So obviously we want oxygen, so it will let oxygen come in to the central nervous system. We want nutrients, it will let nutrients come in. Okay? But it will prevent bacteria from coming into the nervous system. So for example, when there is a damage to this blood-brain barrier, one of the diseases you could get is encephalitis, right, or encephalitis, where you get a reaction in the brain because of a substance that's not supposed to be there. These processes secrete chemicals that promote very tight junctions that form this blood-brain barrier. Okay? As the name suggests, it's a barrier between the blood and the brain. Here's some results from our lab looking at astrocytes. You can see they have this beautiful, they call astrocytes because they have this star-shaped appearance, and you can see that here. You can see that very clearly here. And in this example here, you can actually see them forming the blood-brain barrier around these blood vessels. There's one right at the top there as well, and quite a few over here. Right, so all these structures that appear yellow in this figure here are your astrocytes, because we're looking at two different proteins, so co-localization of two proteins, red and green, give you the appearance of yellow. Another type of neuroglial cell in the central nervous system are the ependymal cells. These cells are important. They line the ventricles. We'll talk about ventricles very shortly. They're holes that are located in the brain, and these holes are very important. We'll talk about it very shortly. Ventricles as well as in the central canal. So in the spinal cord, right down the middle, you've got this, about the size of a pin, structure going all the way down, a hollow tube going all the way down, right? Very small. it contains cerebrospinal fluid. Okay? So these ependymal cells form what is known as the choroid plexus. They line the ventricles, and they secrete from this choroid plexus, they secrete cerebrospinal fluid. Cerebrospinal fluid is very important. It provides protection. It's like a shock absorber, for example. Right? We'll talk about this shortly. And it also carries nutrients. Two other types of neuroglia are the microglial cells and the oligodendrocytes. The oligodendrocytes are the ones that form an insulating sheath around the axons. Okay? So in that video clip as well, you saw the axon with those little tic-tac-like structures around them. So those are the myelin sheaths. Okay? So microglia, they are like the phagocytes that you get in the periphery. They're very similar to phagocytes in function. They're there to protect the central nervous system. They can clean the debris in the central nervous system. So if there's a dead or dying cell, they can come in and actually clean the cellular debris. They can remove waste products as well as pathogens. So if there's damage to the central nervous system, what you see is an upregulation of these microglial cells. Okay? These cells can't proliferate in the nervous system, so they're not like the neurons that do not proliferate. They do proliferate in the nervous system. There's a suggestion, and I'll show you a video shortly, the suggestion that these microglial cells, they sit around, and at least once every minute, they send out processes, and they touch neurons close by. And presumably, that's to check. It's just asking the neurons, are you okay? Okay, as soon as they pick up a signal that's not supposed to be there, you'll find them becoming activated, and they try to insulate the insult that's occurring in the nervous system. Here's more results from our lab. These are motor neurons in the facial nucleus. That's a cranial nerve located in the brainstem. And what we have done in this example is we've actually cut the axons. If you think of this as being in the brainstem, right? And these axons have come out, and they innervate the face, the muscles of the face. So we've actually cut the axons outside the central nervous system. But because we've damaged those axons, what happens is those microglial cells now move in, and they surround these axons, okay? Because those, surround these cell bodies. These are the cell bodies. The axons are out of the nervous system, okay? The axons have come out here. So we cut it as it exits what's referred to as the stylomastoid foramen. We cut the nerve, and this is the reaction that you see. So you can see they come and they isolate these neurons because these neurons have been damaged, presumably doing two things. One, giving this neuron protection so that it can regenerate. Remember, this is a peripheral nervous system neuron. Okay, so the cranial nerve is part of the peripheral nervous system, just like the spinal nerves. So it's giving these cell bodies room to actually regenerate. And these cell bodies do regenerate. The axons do regenerate and make connections with the musculature. So here's what they actually look like. They look like little spiders, right? This too is from our lab. This is in a brain that has been stressed, heavily stressed. You can see that there is an upregulation of these cells. So all these black cells that look like spiders in the system there, these are all microglial cells being activated. Here's one from a European molecular biology lab showing you what you'll see is that these guys lie around there. I think they're green, if I'm not mistaken. And you'll see small processes coming out of them. And what they've done here is they've used a laser and caused a small lesion in the brain tissue. And over a period of, I think it's 180 minutes, so 3 hours, you see these microglial cells swarming in, trying to clean up all the cellular debris. So watch. I don't think there's audio on this. So those green are the microglial cells. Now notice the damage is going to be here. So over a period of 180 minutes. Notice that. So these microglia cells do actually move within the nervous system, trying to clean the system up. Neuroglia of the peripheral nervous system. One is the Schwann cell. Actually, I should go back, sorry. So the oligodendrocytes are the cells that myelinate central nervous system axons. Okay, oligodendrocytes myelinate central nervous system axons. And as you can see, one single oligodendrocyte myelinates one segment of different axons. Okay? So one oligodendrocyte in the central nervous system myelinates one segment of different axons. In the peripheral nervous system, the myelination is done by Schwann cells. Schwann cells myelinate one segment of one axon. That's the major difference between the two, other than their name. So Schwann cells myelinate in the peripheral nervous system, and they myelinate one segment of one axon. And as you can see, what these cells do is that they start by wrapping around the axon, okay, and then they keep pushing their cytoplasm around the axon. So the cytoplasm consists of the lipid bilayer, not the cytoplasm, sorry, the cell membrane. The cell membrane consists of the lipid bilayer, and that's why you see this becoming so white, okay, because this is phospholipids around here. And as you can see, the cytoplasm and all its organelles are in the outermost layer. Satellite cells are another type of cells that actually provide protection to the neuron cell body in the periphery. There are two types of myelination that can occur. Okay. Well, I shouldn't say two types. There's one type that occurs. So there's myelinated axons, okay, and there's unmyelinated axons. Now, unmyelinated axons, the major difference is that they don't have this repetitive structure of insulation. So they're not insulated. Okay? What I like to call this is a Schwann cell giving surrounding axons a group hug. Right? They just kind of keep them in place. They do not insulate them. Here, on the other hand, is insulation. This is like taking tape. If you have a broken cord, you take tape, and you don't just put one wrap around it, do you? You keep insulating it quite a few times, right? And that's to actually help the conduction of the action potential. That's why this myelination occurs, okay, to help the conduction of the action potential. Myelinated axons can conduct action potentials, the message, anywhere between 30 to 120 meters per second. Okay, 30 to 120 meters per second. Unmyelinated axons conduct at around 2 meters per second or less. It takes a very, very long time. Why are these important? Why do we need unmyelinated in the system? Well, very quickly, an example. You stand on a thumbtack or a nail. Okay? What do? Keep pressing down? See how much pain you can actually endure? No. You pull your foot away, right? That immediate reaction that you get is conveyed by these myelinated axons. Okay? That immediate reaction you get is conveyed by the myelinated axons. A couple of days later, you realize that your foot is still throbbing. And you go, okay, I better take a look. And you look underneath, and it's starting to turn black. Right? That information, that throbbing pain that keeps signaling for a long period of time, that's carried by these unmyelinated axons. As long as the stimulus is present, these neurons will keep firing. Okay? So this gives you that immediate signal. This gives you a prolonged signal as long as there's damage there. And the damage basically is, rust from the nail has set in there and causing inflammation and probably gangrene. Okay? Now, big difference between these two and demyelinated axons. Demyelinated axons refer to axons that have been fully myelinated, but through a disease process have been demyelinated. The myelin has been removed. Okay? So antibodies, for example, can attack the myelin, can attack the Schwann cells, or can attack the oligodendrocytes and demyelinate. So big difference. Demyelinate and unmyelinated. Do not confuse the two. They're very different. Now, in the myelinated axon, you'll notice that there are these segments, right? These interruptions here. Those interruptions are referred to as nodes of Ranvier. And they're very important in conducting the electrical potential down the axon. Okay? They're extremely important in carrying those electrical potentials down the axon. They contain a high concentration of ion channels at those nodes, at those gaps, and that's what helps conduct the action potential. Let's just talk about the Schwann cells myelinating. We've spoken about this already. And here, from an electron micrograph, you can see the plasma membrane wrapping around multiple times around that axon. So that's a myelinated axon. And here's an unmyelinated axon. You can see numerous axons in there with one Schwann cell wrapped around it. You don't see that repetitive pattern of myelination. Okay. Questions? All right, moving on to the ventricular system. So the ventricular system in the brain consists of an interconnecting series of channels and chambers. It's derived from an area referred to as the neural tube. When the embryo is developing, one of the earliest divisions of the neural tube forms these, the ventricular system. And what they are, as I said, are actual holes in the brain. Okay? So you will see these holes. I don't think I have an image of this. No. You will see these holes in the brain. We'll talk about them shortly. So in each hemisphere, you have what's referred to as a lateral ventricle. It's kind of a C-shaped structure. It's a little bit more than a C, right? Oh, come on. So this is the ventricle on one side, the lateral ventricle on one side, and this is the lateral ventricle on the opposite side. Okay? This is looking at an anterior-posterior view. If you look at it laterally, you'll see that C-shaped structure I spoke about. Okay? So it consists of, this is the lateral ventricle, consists of the anterior horn of the spinal cord, of the lateral ventricles, the anterior horn of the lateral ventricles, the body, or the genu, G-E-N-U, of the lateral ventricle. There is a posterior horn of the lateral ventricle that actually goes somewhat into the occipital lobe a little bit, right? So it sticks out a little bit. And then an inferior horn of the lateral ventricle. Okay. Now, depending on how CT and MRI cuts are taken, you can actually see these in the MRIs and CTs. So it becomes very important because these become landmark structures when you're looking at CTs and MRIs, right? Because depending on how you cut it, so if, for example, if you cut it like this, okay, you'll see this little bit of the lateral ventricle. You'll see these other ventricles here. We'll talk about it shortly. But you might not see the inferior horn of the lateral ventricle because it's going down in that direction. If you cut it in this direction, sorry, come on. If you cut it in this direction here, now you'll see the anterior horn as well as the inferior horn, and you'll also see what's referred to as the third ventricle. Okay? We'll see this shortly in some MRIs. So the two lateral ventricles, they empty into a midline structure referred to as the third ventricle. Okay, so two lateral ventricles, they empty into a midline structure referred to as the third ventricle. The connection between the lateral ventricles and the third ventricle is referred to as the interventricular foramen. Foramen refers to an opening, right? So here's your interventricular foramen, one on one side, that's on the other side there, underneath. And on the lateral aspect, there's your interventricular foramen. Here is your third ventricle. Okay? So midline structure. The third ventricle then empties into the fourth ventricle via what's referred to as, in this book, the cerebral aqueduct. I actually hate that term. It's actually referred to as the mesencephalic or mesencephalic aqueduct. The mesencephalon means midbrain, and this structure is located in the midbrain. So mesencephalic aqueduct. And then from the fourth ventricle, and notice the landmark of the fourth ventricle, we talk about all the other landmarks shortly. The landmark of the fourth ventricle, the roof of the fourth ventricle is the cerebellum. Okay? And the floor of the fourth ventricle is this area here, referred to as the pons midbrain structure, or midbrain pons. Now from the fourth ventricle, the cerebrospinal fluid then flows around the brain, okay, via the lateral aperture. You can see this here, so it comes out and it flows around the brain, as well as the median aperture. As I mentioned earlier today, right down the midline, the middle of the spinal cord, you find the central canal, a small opening that contains cerebrospinal fluid. Here is a cast, a resin cast of an actual ventricle. So they've actually poured the resin into the ventricles and let it set. As you can see, it comes out with a very similar structure. Okay, I'll leave that to you to identify the different structures in there. So just to give you an orientation on sagittal section of a brain. So the lateral ventricles are inside there. So I don't know whether you can see this here, but there's tissue here referred to as the septum pellucidum. Septum pellucidum. And that's the tissue that actually separates the lateral ventricles, right? So it's tissue that actually separates, as well as other brain tissue. But this prevents the fluid from the lateral ventricles just flowing all over the brain, right? So the fluid is basically inside there. So there's like a hole inside there, in this area there. It flows through the mesencephalic aqueduct into this region here. So this region here, in front of this tissue here, is the third ventricle. So that's your midline structure. Okay? So your lateral ventricles are behind there. That's your third ventricle there. Here is the mesencephalic aqueduct. Okay, so there's actually a pin-sized hole under this tissue right there. And then here's your fourth ventricle. The fourth ventricle has somewhat of a diamond-shaped appearance when you're looking at it anterior-posteriorly, so you can actually see that. Okay, so here's the cerebellum, and there's the structure referred to as the pons, and that's your fourth ventricle right there. This gives you the location of different structures that can be identified on CTs and MRIs. This is obviously tissue that has been sectioned. It gives you the location of where this section originates. So this section here that you're looking at originates around that area there, which is midbrain. Okay, so these are all the structures that you can see, and what you can see here is the mesencephalic aqueduct. Okay, this is midbrain region. That's the mesencephalic aqueduct going through there. That's all midbrain structures. Okay, and then as you come down here, So this is all midbrain over here. We'll talk about this shortly. As you come down here, over this region here, lies your cerebellum. So this then is your fourth ventricle. Okay? All these structures can be identified by MRI and CT. That's just a summary that was in Crosman and Neary. I put that in there. It kind of helps tie it all together. Know only that and you will fail. Here is, as it says, the superior aspect, so looking at it from this angle, okay, horizontally cut. You can actually see the two lateral ventricles, okay, and inside the lateral ventricles you can actually see those ependymal cells, the choroid plexus. These are the cells that produce the cerebrospinal fluid, okay. So here's an MRI showing you the lateral ventricle, okay? The third ventricle, you can actually see the mesencephalic aqueduct right there. You can see a bit of fluid coming in here, so this is all fluid in white, obviously. And there's your fourth ventricle, okay? And you can see the cerebrospinal fluid flowing right around the brain. Now, you continuously produce cerebrospinal fluid over the period of a day. You produce quite a bit of cerebrospinal fluid during the day. So if you keep producing cerebrospinal fluid and don't remove it, what's going to happen? And this happens when you have blockages in this ventricular system as well. Well, the fluid cannot get out. You've got rock or whatever that is, right? You've got a skull. The skull does not move. The only thing that can move is what? The brain tissue. So the brain tissue starts getting squashed and squashed and squashed as the fluid starts increasing inside, and you will eventually die. Okay, especially if the fluid starts pushing the structures down here in the brain stem region. If it starts pushing it through the foramen magnum, the opening at the top of the spinal cord, you will die because we'll talk about the structures located there in the brain stem region. You have structures that regulate respiratory function, for example, cardiovascular function. Okay, so when they get squished and pressed, you will die. So cerebrospinal fluid produced in the lateral ventricles flows through the interventricular foramen into the third ventricle. You can see the third ventricle is that midline structure. Through the mesencephalic aqueduct into the fourth ventricle, through the lateral and median apertures into circulation around the brain. And then, via these structures referred to as arachnoid villi, that actually, this is actually the meninges protruding through into a sinus, right? So right down the middle, you've got the superior sagittal sinus running right down the middle of the brain. That's the drainage system for blood. Okay? The cerebrospinal fluid, because it's under such high pressure compared to blood, will push CSF into the blood. This is the venous return, right? The sagittal sinus is the venous return, and that's how you get rid of the CSF. Okay, so this happens continuously throughout the day. So blockage in any one of these systems, so if you think of a blockage here in the mesencephalic aqueduct, if it's blocked there, what's going to happen is these structures will start expanding. Okay, as these structures start expanding, as the ventricles start expanding, the brain tissue will start getting squished against the bone, against the skull, causing damage. Questions on that? Okay, cortical and subcortical structures. We spoke about some of these structures a couple of weeks ago. The central sulcus, the precentral and postcentral gyri, precentral associated with motor, postcentral associated with sensory. We have the lateral fissure, also known as the Sylvian fissure. Okay, okay, separating the frontal area from the temporal lobe. We have the superior temporal gyrus, the middle and the inferior temporal gyri on the lateral aspects of the brain. On the more medial aspects, we still have the central sulcus. We have the precentral and the postcentral gyri. We also see a division between the parietal lobe and the occipital lobe, referred to as the parieto-occipital sulcus. We have the calcarine sulcus, and on either side we have the primary visual cortex. We also have the cingulate gyrus. We know its function. We've spoken about its function. And just inferior to the cingulate gyrus, we have this band of white matter, referred to as the corpus callosum. Corpus callosum. So as you can see, this is a major band of white matter. And what it does is it connects the right hemisphere with the left hemisphere. Okay, so these are axons that are going in both directions, helping the one side of the brain talk to the other side of the brain. The structure here referred to as the parahippocampal gyrus. We'll talk about this shortly. The hippocampus is associated with memory. Parahippocampal gyrus also contributes to being part of the hippocampus. And then the uncus here, associated with smell. We'll talk about that a little later. Now talking about some deep brain structures. The basal nuclei. Now, I need to point out here that around 99.5% of the textbooks, there are only 2 textbooks I've read so far that actually call these basal nuclei. Most of the textbooks call these basal ganglia. Anyone have any idea what the problem with that is? Absolutely, well done. It's in the central nervous system. It's a collection of cell bodies in the central nervous system. We said that collections of cell bodies in the central nervous system are known as nuclei, right? So calling these basal ganglia is wrong. Okay? So we will not perpetuate that mistake, but textbooks still call them basal ganglia. So note that. So these are 4 subcortical structures located deeper within the brain, and they play a major role in normal voluntary movement. Okay, so damage to some of these structures, such as the substantia nigra, can cause Parkinson's disease, right? Damage to other structures can cause chorias. This is, you see people, they're standing there and they'll do this, right? That's called a choria. It's called a choria because it's called choreiform, which is like a dance choreography, right? And you'll see they have contorted structures, the facial, orofacial structures as well. So these nuclei are heavily involved in normal voluntary movement and also in tone of the muscle. Interestingly, they do not have direct input or output to and from the spinal cord, right? So, for example, if they're controlling normal voluntary movement, you'd think that they have direct control of the spinal cord. They don't. They exert their effects by communicating with the cortex, where the information, the motor information actually originated. Okay, so it's kind of a loop-like structure. So what happens is you receive sensory information, and the cortex now needs to perform a task. What the cortex will do is send information to the basal nuclei saying, this is the sensory information that was received. This is my plan to perform that action. Right? The basal nuclei will then say, well, okay, that plan, this was the sensory information. Well, that plan's not quite right. Fix it. And that loop will go back to the cortex. Cortex will try to fix it, come back, and it'll go in a loop. How long does this loop take? A couple of weeks? Seems like that when I ask questions because it takes forever to answer. But it doesn't. It's quick. It's rapid, extremely rapid. Okay? So these basal nuclei, as well as the cerebellum, they kind of act as consultants. Okay? So you've seen how the cerebellum works, right? Works really well, works really poorly on Friday nights or Saturday nights, right? Cerebellum is involved in coordinating movements. They also have motor plans stored in there. Okay, and they love alcohol. They like to party too. So the primary input to these basal nuclei is from the cortex. And the output is via the thalamus. This is a structure, this is a group of nuclei, we'll talk about it later, located on either side of the third ventricle. Okay, so third ventricle is a midline structure. On either side you have two thalamic nuclei, groups of cell bodies. So via the thalamic nuclei, they then send information to the prefrontal, premotor, and motor cortices to coordinate that voluntary movement. So here's what these look like. They too have kind of that C-shaped appearance. Very important. You'll see why on the next slide. So here's your midline thalamus, two thalamic nuclei. Okay? And on either side, you have your basal nuclei. So you remember this first one, which are your ventricles. Okay? You know your lateral ventricle, which goes all the way around. So all this is lateral ventricle. Following the lateral ventricle all the way around. Oh, actually, okay. So that's the lateral ventricle. That's your third ventricle, the midline structure. And then on either side of the third ventricle, you find the thalamus, two thalamic nuclei. Following the lateral ventricles all the way around, except into this posterior bit, is what's referred to as the caudate nucleus. Okay, so it follows it all the way around. Why is this all important? You'll probably see in the next couple of slides, because you can see these very nicely in MRIs and CTs. Okay? You'll actually see that the caudate nucleus, this area here is referred to as the head of the caudate nucleus, the body, and the tail of the caudate nucleus. So depending on how you cut, how your MRI sections are taken, right? You might actually see, you will see the head of the caudate nucleus, but you might also see the tail of the caudate nucleus. And the caudate nucleus actually ends in the structure referred to as the amygdala, which is highly involved in emotional behaviors. The most lateral aspect of these basal nuclei is this group of this nucleus referred to as the putamen. The putamen. Right? So you have two nuclei on each side, one on each side, referred to as a putamen. So here's an attempt at giving you kind of a 3D view of this, if at all possible. You have the putamen on the most anterior aspect. Right at the head, right at the most anterior aspect of the putamen, the caudate and the putamen are kind of one. Okay? And then as you go posteriorly, the putamen stays midline. The putamen stays midline and the caudate kind of pulls away. And you'll see this in the images that come up shortly. More medial to the putamen is another nucleus referred to as the globus pallidus. Globus pallidus. We won't be going into the details of the circuitry of how these basal nuclei function and how they produce movement, and without them, how you have deficits in movement or even no movement, depending on how bad the injury is or the damage is to these nuclei. But these structures are all intimately connected. So information travels from the putamen to the globus pallidus, from the globus pallidus to another nucleus, then back to the thalamus, and then out of the thalamus. But we won't go into that detail. And then down here in the midbrain region is the last of the basal nuclei, substantia nigra. Okay? The substantia nigra consists of two parts. One referred to as pars compacta, because the cells there are highly compacted, and the other part referred to as pars reticulata, because the cells there appear to be in a network-like structure, reticulum, right? And it's the pars compacta cells that actually die in Parkinson's disease. The pars compacta cells produce dopamine, and dopamine is critically important for movement, for the smooth movement. Okay? Without that dopamine, you have the kind of jerky movements or the inability to move if there are very, very low levels of dopamine. Okay, so here's what a coronal section looks like. Here are your ventricles, your two lateral ventricles. Okay, there's your third ventricle right there. Following the lateral ventricles all the way around is the caudate nucleus. So you can actually see, I'll show you some MRIs, where the caudate nucleus actually impinges on the lateral ventricles. It actually protrudes into the lateral ventricles and follows it all the way around. Okay. There's your putamen. Okay. So at the most anterior bit right here, as you can see it, they show you here, the caudate and the putamen are actually one. And then as you start moving posteriorly, they start separating. The caudate pulls away from the putamen as you go posteriorly, right? So there's your putamen, and here's the globus pallidus. You'll notice that in the previous slide, they use lateral and medial, okay? But most textbooks and most clinicians use the term interna and externa. Okay, externa referring to the lateral part of it, interna referring to the medial part of it. There's your third ventricle, and on either side of the third ventricle are your thalamic nuclei, one on each side. Just below the thalamic nucleus is the subthalamic nucleus. And in the midbrain region, you find your substantia nigra. Two compartments to it, pars compacta, pars reticulata. And here's what you can expect to see on your exams, right? You can expect to see MRIs or CTs with arrows pointed towards it. Now you see the set of lecture notes that I just released over the weekend. Right at the end of it, you have about 20 MRIs, okay? That's for your study references. So when you actually look at the structures, go to those figures and try to identify where these structures are, okay? One important thing I forgot to mention here is between the caudate nucleus and the putamen, and between the thalamus and these basal nuclei, all this area here is all white matter. All this is white matter. Okay? What happens is that all this white matter comes together and travels through this region here. This region here is referred to as the internal capsule. Internal capsule. Here. Right? Internal capsule. If you can pick where not to have a stroke, pick the internal capsule. Why? Because I just told you that all the fibers, all the axons from the cortex going down to the spinal cord travel through the internal capsule. So those are your motor fibers, right? All your sensory fibers going up to the cortex also pass through the internal capsule. So if you do have a stroke in the internal capsule, you'll have both sensory and motor loss on the contralateral side of the body. Okay, we'll talk about these crossing fibers shortly. So you'll have both sensory and motor loss on the contralateral side of the body. So here's an MRI showing you these images. So here are your lateral ventricles. You can see the two lateral ventricles. You can see the septum pellucidum, the thin layer of tissue that separates the lateral ventricles. Protruding into the lateral ventricle, you can see the head of the caudate nucleus on both sides. You can see the internal capsule. This is referred to as the anterior limb of the internal capsule, the genu or body of the internal capsule, and then the inferior limb of the internal capsule down here. Here's your putamen. You can clearly see that. There are your thalamic nuclei. Okay. Here's the insular cortex I referred to in the lecture a couple of weeks ago. I said you can't actually see it because it's underneath the sylvian fissure. It's right. If you separate it, you might be able to see it, but here's your insular cortex. Right, so here's your sylvian fissure there, and there's your insular cortex. We'll talk about the fornix later on today, I think. Down here, right, this is the inferior horn of the lateral ventricle. So now you can picture the way it's been cut. And there, in there, you see the tail of the caudate nucleus right there. Okay, so this is the inferior horn of the lateral ventricle, and there's the tail of the caudate nucleus right there. So there's a horizontal section, there's a coronal section. I'll let you identify the structures in there. They've been identified above as well. Here's another section that actually shows you all the structures we just spoke about, but also shows you the globus pallidus. You cannot see, or you might be able to see the two, the externa and the interna. There's the externa, there's the interna right there. Okay, and this structure here, you can see the hippocampus as well. It's the tail of the caudate nucleus. It says the temporal horn of the lateral ventricle, or the inferior horn, depending on what you want to call it, together with the choroid plexus. Okay, so what I've tried to do here is taken two different sources, and I've tried to show you an area from where these images might have been produced. This is anterior, quite anteriorly, over there. And as you can see, the caudate and the putamen, you can actually see the fibers separating as the putamen pulls away from the caudate. Right? You can see the fibers separating it right there. You can see them running through there. And the internal capsule now starting to come through. Okay, you can see that right there as well. Okay, let's take a 5-minute break. We'll come back for the next bit. All right, so I'm going to try and get through as many of these slides as possible, but please remember that there's an extra lecture that's going to be placed online. So in addition to the lecture, I think next week, right? We still have one more lecture next week. There's another lecture that will be placed online, and that will deal with all the neuropathology and associated diseases. Okay, so, and also remember, I'll just wait till the rest come in to actually mention this. So we'll talk about the limbic system here. Limbic system is highly associated with emotional aspects of behavior. Okay, and parts of the limbic system include the cingulate gyrus, which is actually sometimes referred to as the limbic lobe, the cingulate gyrus, the hippocampus, the parahippocampal gyrus. Now note that the parahippocampal gyrus actually lies over here. Okay, it's been kind of shaded out just so that you can see the hippocampus and see the deeper structures. So remember that. Something I forgot to mention earlier that you will actually come across is when you look at some CTs and MRIs, especially if you look at it on a sagittal aspect or looking at it laterally, what you will see is that in the third ventricle, you see a small hole in it. If you go back and look at the figures from the lecture, the notes on the ventricular system, you'll see a small hole in the third ventricle. That hole is caused by this structure right here, referred to as intermediate mass, or massa intermedia. Now this intermediate mass comes out of the two thalamic nuclei, but you would think that they actually have connections running through, right? Like the corpus callosum, they have axons running through. They don't. Okay, this function is not really understood right now, but we just know that they come from the two sides and they stay juxtaposed like this. But that causes that little cavity that you see in the third ventricle.
Is that the same thing of the anterior and posterior commissure?
They are different. Anterior and posterior commissure, we'll get to that shortly, they are actually connecting bundles. Okay, so they are fibers that actually go across just like the corpus callosum. Okay, we'll come to that shortly. But this is not. The fibers do not go, they do not run through. So they do not go from one hemisphere into the other. They just come out like this, just like a piece of, tissue juxtaposed like this. But that causes a very characteristic hole-like structure in the third ventricle. So go back and look at that, and you'll actually see it. So here's the limbic system. Oops, what happened there? There's the limbic system highlighted for you. And as I said, cingulate, parahippocampal gyrus, hippocampus. There are some other structures that are also associated with this, but we won't go into that detail. We'll leave it at that. The amygdala, as I mentioned, lying right at the end of the caudate, right at the end of the caudate nucleus putamen. Okay, it plays a significant role in the processing of memory, and especially memory associated with emotion. Okay, so damage to the amygdala, and you can see this on both sides. Where am I? Right, so these are the amygdala nuclei. This is looking at the anterior-posterior view. This is at the lateral view. Damage to these nuclei can cause significant emotional disturbances in individuals. The hippocampus, the hypothalamus. The hypothalamus is another structure. I think we'll go into a little bit of detail about the nuclei in the hypothalamus. It is responsible for controlling numerous functions, okay, including the regulation of body temperature, satiety centers for hunger, thirst, etc., regulates circadian rhythms, and plays somewhat of a role in emotional behaviors as well. The hippocampus, we have one on each side, just to give you a view of the hippocampus running through, plays a very significantly important role in memory consolidation. Right? So all long-term memories are stored in the hippocampus. So you can have depending on the damage to the brain, you can have selective memory loss. You can lose long-term memory, or you can lose short-term memory. Okay? Which one's better to lose? Neither. Right? But unfortunately, structures such as the hippocampus are the first that are affected in diseases such as Alzheimer's disease. Right? It starts in the hippocampal region, so the first signs of Alzheimer's or dementia is usually the loss of memory. Right? People start losing memory, and then their loved ones go, oh, this is not good, let's go get it checked out. The problem with all these diseases is that by the time those deficits start showing, the memory deficit starts showing, it's believed that you've already had this disease for about 10 or 15 years. Okay? So that's the problem that are facing clinicians and even scientists right now, the fact that it's too late. By the time the deficits are actually identified, the damage has already been done. You've lost significant amounts of neurons in these regions, and as we know, neurons are post-mitotic. They do not divide. New neurons do not form. There was a long-held belief that the hippocampus was probably the only region in the brain where neurogenesis did occur, so new neurons did form. But just about a month ago, 2 papers came out, very strong papers, suggesting it's all false. It doesn't. So the jury is still out, because these papers have just come out, saying that there's no neurogenesis in the hippocampus. The hippocampus is a very beautiful structure. Okay, it has this kind of interconnected C-like structures, very beautiful cytology, cytoarchitecture. So this is a human hippocampus here, right? Showing you all the different regions, including part of what's referred to as the entorhinal cortex right there, the subiculum, and these regions right here. This is in a rodent, this is in a rat or a mouse. And what's really nice is what you can't see here is the cytoarchitecture of these cells. There are very different cells, and that's why these structures have very different names, because the cells in these different regions appear different, and they have very distinct functions, which we will not go into. Limbic system obviously is very important. As I said, it establishes emotional states. It facilitates memory storage and retrieval, and it also provides conscious links to functions. So you can actually consciously associate certain emotions with certain stimuli. Okay, components of the limbic system. So that was just a blow up if you have to go back to it, if this thing is too small. Numerous structures in here. The amygdaloid body. The amygdaloid body actually links the limbic system with the cerebrum and other sensory systems as well. The limbic lobe consists of the cingulate, the dentate, the parahippocampal gyrus, and the hippocampus. So these structures are all shown here, right? You can see the amygdaloid body. Here's the hippocampus. And within that hippocampus is the dentate gyrus, and the dentate gyrus refers to this region right here. The region right there. That little bit is called the dentate gyrus right here, DG. Okay. The fornix is a tract of white matter, and now you see this if you go back to a few slides looking at the MRI, you'll actually see this fornix. It's a tract, so it's white, so white matter, so it actually, you can see it, that connects the hippocampus with the hypothalamus. Okay? So you can hopefully see now how emotionally related functions can, by going through the hypothalamus and looking at the functions of the hypothalamus, start associating different behaviors. Just because I'm skipping does not mean that it's not important, right? Probably means it might show up on the exam. Reticular formation is a group of nuclei located mainly it starts in the midbrain region and travels all the way down right up to the spinal cord. Reticular meaning network. It's believed to consist of around 100 nuclei doing different functions. Okay? And some of the functions are described here. But we'll talk later about how the reticular nuclei are also important in, as I mentioned before, cardiorespiratory functions, cardiovascular functions, okay, satiety, numerous different behaviors. Once again, know only this and you will fail. Just a brief introduction to the development of the brain itself. So as the embryo starts forming, one of the first things that you start seeing is the development of this nervous system as the different layers of the blastocyte starts forming. Know the different primary brain vesicles. We won't go into the details. Know the different primary brain vesicles, and from them you get the secondary structures, and then the structures of the mature brain. So the prosencephalon leads to the formation of the secondary structures of the telencephalon and the diencephalon. Okay, so the prosencephalon, the forebrain, and you can see here how the formation of these different structures. So here's your prosencephalon. Here's your mesencephalon or midbrain. Okay, and then the rhombencephalon. Notice how I mentioned right at the start, I said, you know, in bipedal animals there are different flexures in the nervous system, and that's why the terminology actually changes. Okay, so you can actually see very early on, you can see the cranial and the spinal nerves starting to develop. Right, as you see here, you can see the eye bud starting to form, the optic vesicle starting to form. And then as you progress, so the mesencephalon, its secondary structure remains as the mesencephalon. Okay, so if you're looking at it here, so notice now that the prosencephalon has developed into a telencephalon, which will develop into the brain, and the diencephalon, which will develop into the thalamus. Then your mesencephalon stays as mesencephalon, develops into the midbrain. And your rhombencephalon then develops into a metencephalon and a myelencephalon. The metencephalon going on to develop into the pons and the cerebellum, and the myelencephalon forming the medulla oblongata, the medulla. And we'll go into those structures shortly. The diencephalon, which once again develops into the thalamus, is very important because it integrates both sensory and motor information. So all information that goes up, which is sensory information, arrives at the thalamus. The thalamus acts as a gating center. So the thalamus decides whether that information is important or not. So some of you have started nodding off, and that's your thalamus telling you that this lecture is not important. The thalamus is wrong in that case, right? And then, as I mentioned before, I said that all the information from the cortex, motor information, travels to the basal nuclei. From the basal nuclei, they go through the thalamus back to the cortex. They do not go directly from the basal nuclei to the cortex. So the thalamus acts as a gating center, right? So all the information, the sensory information goes there, and the motor information goes there. So this shows you now the adult system. So here's your thalamus, thalamic nuclei. Here's your midbrain or mesencephalon. Here's your metencephalon right there, right? And then your medulla or the myelencephalon. So these are the adult structures, and these are the names that we will be using. So we used thalamus, we'll use the term mesencephalon, midbrain interchangeably. The pons, we will call the pons. And the medulla, we'll either say medulla or medulla oblongata. The diencephalon consists of these three structures, the thalamus, the epithalamus, and the hypothalamus. As I mentioned before, the thalamus itself is made up of numerous nuclei, groups of cells, associated with very different functions. Okay, so here's the input, as you can see there, and there's your output going to the different systems in the cortex. Okay, so here's, for example, visual input comes to the structure referred to as the lateral geniculate nucleus. General sensory information arrive in this ventral group of nuclei. Information from the cerebellum and the basal nuclei to very distinct regions within the thalamus. Another important structure is the pineal gland. It is found in the epithalamus, the most posterior aspect of the epithalamus, and it is the gland that's associated with secreting the hormone melatonin. Right? Melatonin is important in numerous functions, and one of the functions is the sleep-wake cycle. So individuals who are traveling long distances, rapidly going back and forth, they actually try to supplement their system with melatonin so that they can actually adjust their body clock to the country that they're going to visit, right? So that you feel sleepy at the right time and feel awake at the right time. You can actually get calcification of the pineal gland, which obviously will stop producing melatonin when it's calcified. There are other diseases that are associated with that. The thalamus, again, another very important structure. Now this figure here, is it in your notes? Yeah, okay, good. Couldn't remember that I put it in before or after. This is, once again, taken from those 20-odd images right at the back of the lecture, right at the end of the lecture, right? Just showing you different structures. So be able to not only identify structures, right, but be able to associate function with structures as well. And once we do the blood supply, you will have to be able to associate the blood supply to the different structures as well. Okay? So exam questions could say, what is this structure? Where does it receive blood from? What is a disease associated with this structure? Or damage to the structure will cause, and you'll have to pick the disease that it might cause. So here's your thalamus on either side. There, that's your lateral ventricle there. This is the inferior horn of the lateral ventricle. And there's your third ventricle down there. And on either side are your thalamic nuclei. Oh, well, here, okay, I didn't realize this was in there. So here's that structure I was talking about, massa intermedia. And as you can see, in the third ventricle, you actually have that hole in the third ventricle caused by massa intermedia. So different groups of the thalamic nuclei. The anterior group is associated with the limbic system, so therefore associated with emotional aspects of behavior and memory. You can read this, right? I'm going to skip through this in interest of time. The posterior group, important, is a pulvinar nucleus associated with sensory information. The lateral geniculate with visual, and the medial geniculate with auditory information. Okay, so these act as relay centers for the information. Oops. Hypothalamus, very important structure. As you can see, numerous nuclei in the hypothalamus, right? Numerous nuclei in the hypothalamus. From this region of the hypothalamus, you have a structure that hangs out of that, referred to as the pituitary gland. I'm sure you know how important the pituitary gland is. So structures of the hypothalamus, the mammillary bodies located here, processes olfactory and other sensory informations, also controls reflex eating movements. So damage of the mammillary bodies can have significant effects. Now these mammillary bodies, as you can see, they actually protrude slightly out of the tissue, so you can actually see this. Okay, you can physically see this both on the tissue as well as on MRIs, etc. The infundibulum refers to this stalk right here. Right, that area there is referred to as the infundibulum. It connects the hypothalamus to the pituitary gland, and it helps in the, obviously, you can see these axons traveling through here, and also helps transport hormones, etc., produced by the pituitary gland. The tuberal area just looks like an area of white matter, just sits around there, located between the infundibulum and the mammillary bodies, and it helps control pituitary gland function. What I should also mention is just anterior to the pituitary gland, okay, is what's referred to as the optic chiasm. The optic chiasm. Now this is a structure that's significantly visible from the inferior aspect, if you're looking at the brain from an inferior aspect. And this is where, I'm not sure whether you know this or not, your nasal hemispheres of your eyes, they actually cross. Did you know that? The lateral hemispheres go to the same side. They stay ipsilateral. The nasal hemispheres actually cross. So half your eye crosses, the other half stays on the same side. The area where this crosses is referred to as the optic chiasm. Functions of the hypothalamus, numerous, controls autonomic functions, provides both subconscious control, or provides subconscious control of skeletal muscle. Notice it says subconscious control, because you can't activate the hypothalamus consciously, right? So therefore, it's subconscious control. Very importantly, coordinates activities between the nervous and the endocrine systems. Very important. If that coordination is lost, obviously the endocrine system will fail to function. Secretes numerous hormones. These are just a few examples. Antidiuretic hormone by the supraoptic nucleus. And oxytocin, very important. Oxytocin, extremely important, by the paraventricular nucleus. Emotional and behavioral drives as well. The suprachiasmatic nucleus also controls circadian rhythms. Okay, so notice all the different structures. And I think during your study you will be trained to actually identify these structures. We're not trying to do that here in this unit of study. And I'm not sure why I stuck this in here, other than to show you, I guess, the wet brain specimen, showing you most of the structures we spoke about today. So here's the cingulate cortex right there, cingulate. Here's the corpus callosum, okay, septum pellucidum, choroid plexus inside the lateral ventricles, the thalamus that normally surrounds the third ventricle on either side, two thalamic nuclei. Here's the hypothalamus, this whole region right here. There's the anterior commissure that you spoke about before, and here's the pineal gland. Here is the optic chiasm right there, so that's where the cross occurs, and here's part of the optic nerve. And right here, what has been cut out is the pituitary stalk, or the infundibulum, right? So that has been cut out because it normally dangles down here. There's the tuberal area, and here, as I said before, you can actually see the mammillary body. Some other structures that I'd like to point out. There's your fornix, connecting the parahippocampal gyrus to the hypothalamus. So you can see here it's coming into the hypothalamus. Here is the, what's the name of this? This opening right here. Sorry? That's the mesencephalic aqueduct, right? Because this area here, right there, is your midbrain. Okay? You can see, I'm not sure whether you can see it over there, but you can see a slightly darkish area right there. I'm not sure if you can see it, but that's your substantia nigra. Okay? Substantia nigra, black substance, or substance black. It's very clear. There's your fourth ventricle with your cerebellum, and here's your pons on the anterior aspect of it. Okay. All right, now moving on to the brain stem. The brain stem, remember, is, especially the medulla oblongata, is continuous with the spinal cord. Okay, so there's no separation basically. It's a continuous structure. The brain stem includes the midbrain, pons, and medulla oblongata. Not the cerebellum. Okay, remember that. So looking at the midbrain, there are quite a few structures in the midbrain that can be identified and that you need to know. What you'll see on the inferior aspect is these four groups of nuclei. They actually protrude, they sit out quite significantly. They are referred to as the superior. We have two superior nuclei, superior colliculi, sorry, and two inferior colliculi. Okay, and you can see these and you will be asked to identify them. Also in the midbrain section, so this area in green here is the midbrain, okay? You find what I referred to as the cerebral peduncles. Cerebral peduncles carry the axons, the motor axons that are actually traveling down from the cortex. So from the cortex, they come together through the internal capsule, and now in the midbrain region, they travel through the cerebral peduncles, and then they travel down, and we'll talk about where they travel to later. And as I mentioned, in the midbrain region, you see substantia nigra, again, two nuclei. And you also see another group of nuclei referred to as the red nucleus, or nucleus ruber, meaning red nucleus. And it's called red nucleus and it's called substantia nigra because you can actually see that it's not stained, but there are pigments in these structures. So substantia nigra actually has pigments in them. It's referred to as neuromelanin, which makes it appear black. And if you take the brain of a person who has had Parkinson's disease and died, and you look at the level of the substantia nigra, you'll see that it just appears clear. Not clear, but it appears like normal brain tissue. You don't see the black, because the dopamine-producing cells, those black cells, have actually died. The neuromelanin-containing cells have died. So these structures here, especially the red nucleus, we won't go into the details of these, but they form, and I'll mention it at the end, they actually form postural motor systems. So they are systems that actually help us stand with very little conscious control. Right? If you have to stand, you stand. You don't actually understand the intricacy of actually just getting up and standing until you have damage to these systems. Okay, so they're very important systems. So this system, starting at the red nucleus, is called the rubrospinal system, because it starts at the red nucleus and travels down to the spinal cord. Rubrospinal system. It's very important in subconscious control of movement. And some believe that it might be involved, this particular system might be involved in the conscious control as well. Although, once again, the jury's out on that. Now, the midbrain itself can be divided into what's referred to as a roof and a flow. The roof is referred to as the tectum. Okay? The tectum, and it consists of the superior and the inferior colliculi. Superior and inferior colliculi. The tegmentum region, I will show you some, I'm sure we'll show you some images shortly. Tegmentum region contains the ventral portion of the midbrain. It's also referred to as crus cerebri. Those two pairs of nuclei that you see are referred to as corpora quadrigemina, because there are 4 corpora quadrigemina. The superior colliculus is involved in visual processing, very preliminary visual processing, as well as the control of eye movements. And the inferior colliculus is involved in forms of auditory processing. Not the primary auditory processing, but some forms of auditory processing. Okay, here you go. So we're looking at this region right here. Here's the optic nerve. There's your optic chiasm right there. Okay, mammillary bodies. These Roman numerals refer to the cranial nerves. We'll cover that very shortly. The cranial nerves. And this region here is your midbrain, right? Very small region, as you can see, just that region there, okay? And on the dorsal aspect, you have the superior and the inferior colliculi. So you see it there, superior, inferior colliculi. And then on the ventral aspect, you have what's referred to as the tegmentum. So that's the tectum right there. The tegmentum runs all the way down. Consists of a midbrain tegmentum and a pontine tegmentum. While I'm here, I'll also show you some other structures. So here we have the cerebellopeduncles. Cerebellopeduncles. These are axons, groups of axons that connect the cerebellum to the rest of the brain. Okay? So we have a superior cerebellopeduncle, we have an inferior cerebellopeduncle, and we can have quite a large middle cerebellopeduncle. Okay, we'll come back to this later. I'm just highlighting it on this slide because it's there. Here then is your mesencephalic aqueduct. There you see your massa intermedia. Mesencephalic aqueduct, 4th ventricle. Okay. It's just I once again identify the different structures. In the 4th ventricle, you can actually see some signs of choroid plexus. Okay, so cerebrospinal fluid can be produced in the lateral ventricles as well as the 4th ventricle. Some in the 3rd ventricle as well. You've got to remember that these are not carbon copies, right? There are variations between individuals and significant variations. The tegmentum consists of the red nucleus and substantia nigra, important structures. It's a multisynaptic network of neurons containing both ascending and descending tracts, and the nuclei of cranial nerves 3 to 12. Okay, that's in the tegmentum, the entire aspect of the tegmentum. We spoke about the cerebral peduncles, these structures here on the side. So note we have cerebral peduncles and we have cerebellar peduncles. Okay, different functions. The pons is a large bulb-like structure that you see on the ventral aspect. Consists of a pontine tegmentum and the basilar pons. The basilar pons is that bulb-like structure that you actually see. Okay, this little bit here. And the pons is very important in linking the cerebellum with the mesencephalon, diencephalon, cerebrum, and the spinal cord. As you can see here, it shows you tracks traveling through the superior cerebellar peduncle. Okay. Here's your middle cerebellar peduncle traveling in that direction there, and the inferior aspect of it. So within the pons you find the motor nuclei of cranial nerves 5, 6, 7, and 8. So once you know what functions these cranial nerves have, you should be able to link them back to damages within the pons, because these are where these are where the cell bodies are located, right? You also have nuclei involved in respiration. You have other groups of nuclei, so cell bodies, as well as tracts traveling through the pons. You have both ascending, descending, as well as transverse tracts. Okay? So that's how the cerebellum can actually communicate with each other, the lobes of the cerebellum. This image is, I think, to show you the different peduncles. So there is a superior cerebellar peduncle. Notice I mentioned that the fourth ventricle, the floor of the fourth ventricle has that diamond-shaped structure, right? So now above all this here, what's being cut out is the cerebellum. Okay, so you're looking at this in this direction with the cerebellum cut out. The middle cerebellar peduncle is quite large. It consists of transverse fibers. The medulla oblongata is the most caudal segment then of the brainstem. So it is continuous with the spinal cord. It extends rostrally. Which direction is that? Rostral? Goes up. Yeah, rostrum means beak, right? So going up. Extends rostrally from the level of the foramen magnum. Right, foramen magnum is the opening through which the spinal cord travels. So all tracts passing to or from the spinal cord must travel through the medulla. So obviously damage to the medulla then, damage to the entire brain is catastrophic, but damage to the medulla then has significant characteristic deficits. Cranial nerves 6 to 12 are associated either, so the nuclei are located either within the medulla or at the pons medulla boundary. And the medullary reticular formation, those 100-odd groups of nuclei. So they contain these groups of nuclei that influence numerous. I've just mentioned two here. They contain numerous functions. There are axons that travel from certain reticular formation nuclei that also control posture. They're referred to as reticulospinal tracts. Okay? So they are involved in almost every single thing. And this is why if you have intracranial pressure building, you need to shunt that out, because or else what will happen is the tissue will start spreading. It'll start spreading this way, and it can't, right? So not tissue, sorry, the fluid will start spreading. The tissue gets compressed this way. If it starts getting compressed this way, you will die. Okay, it's almost instant death. Because it tries to push through foramen magnum, and when it starts impinging on these centers, obviously, life will stop existing as you know it. Okay, so if you look at the medulla itself, it has some very characteristic bumps or humps, whatever you want to call it. Characteristic structures. On the anterior aspect, there is a division that runs through, referred to as the anterior median fissure. Anterior median fissure. And on either side of this anterior median fissure, you get these two very characteristic humps called the pyramids. Okay? Lateral to the pyramids, there are another two smaller nuclei referred to as the olives. Oh, here you go. Okay. So there's your pons. There's your anterior median fissure. And just lateral to the anterior median fissure on each side, you find the pyramids. Pyramids are critically important structures. The pyramids contain the axons of all your motor neurons. So notice we said they start in the cortex, they travel through the internal capsule, through the cerebral peduncles. Now they travel through the pyramids on their way down to the spinal cord. Okay? And then here, on either side here of the pyramids, are the olives. You can't actually see that. That's underneath there. There's the olive right there. Note this region here that says pyramidal decussation. We'll talk about, this shortly. Talk about it now. Approximately 90% of the axons traveling down, so these are the motor axons. Approximately 90% of the axons traveling down cross over onto the opposite side at the level of the pyramids. At this level referred to as the decussation. Decussation means crossing over, right? So at the pyramidal decussation, around 90 to 95% of the fibers traveling down on this side will cross over and innervate the opposite side of the body. Okay? So now this brings in the next step, where if a lesion occurs above or below the level of the pyramids, what side of the body would be affected? Obviously, if it's above the level of the pyramids, what deficits would you see? Contralateral, but what deficits would you see? Motor deficits, right? Motor deficits, these are fibers that are going down, right? Your sensory fibers go on the dorsal aspect of your spinal cord. Your motor fibers travel on the ventral aspect of your spinal cord, right? So sensory going up on the dorsal, motor coming down on the ventral, right? So if the damage is above the level of the decussation, the effect will be contralateral motor paralysis. If it's below the level of the decussation, they make brilliant exam questions. Below the level of the decussation, it's ipsilateral motor deficits. Okay, is that clear? Any questions on that? No? Okay. They're called corticospinal fibers, showing you the origin and the termination. Originates in the cortex, terminates in the spinal cord. The olives, I'll let you read that, consists of two separate nuclei, inferior and superior olivary nuclei, and their involvement, function. The posterior medulla, posterior aspect, is now characterized by another two columns, referred to as the gracile and cuneate fasciculi. Tracts, the fasciculi are tracts, and respective tubercles, which are the little humpy bits that you actually see. Okay? As it says here, the tubercles are formed by the underlying nuclei. The gracile tract contains axons. So what axons would the gracile tract contain then? What type? Sensory. Good, right? Sensory. Gracile. Think about gracile being graceful. Ballerinas are graceful with their feet, right? So tracts below T6 travel through the gracile tracts. Okay? Tracts from the arms, etc., sensory tracts from the arms, etc., travel through the cuneate tracts. It makes sense because if you actually think of designing this system, right, if these are your feet down here and you keep going up to the cortex, it's easier to add laterally than to add medially, right? So as you keep coming up, so this would be your gracile, and as you keep coming up, you add your hands and your upper limbs, etc. So T6 and above is cuneate, below T6 is gracile. So once again, depending on where the damage is, you can have sensory loss either from the lower half of the body. If the damage is high up, involving both the gracile and cuneate, I hope you guys can see the exam questions. Don't say I didn't warn you, right? You guys are not responding, but just remember this. Depending on where the damage is, you could have complete sensory loss from that side. If it's only the gracile, even if it's up here, if it's only gracile, you'll only have sensory deficits from the leg. Okay? If it's both gracile and cuneate, it'll be that entire side. And we'll talk about what functions are associated with these. If it's only cuneate, then it would be only the upper bit part of the body, sensory loss from the upper part of the body. So I usually go with the majority, and usually majority in a class is about 1 or 2. So I see 1 or 2 people doing this. I'm happy. I know they understand. I'll keep going. So if you have questions, make sure you ask me, okay? So here are some of the nuclei located in the medulla, part of the reticular formation. You can already see these now, right? Here is the gracile nucleus, nucleus gracilis, and the cuneate nucleus. Three general groups of nuclei, autonomic, sensory and motor nuclei, as well as the relay stations. Okay? Here's what the relay stations actually relay, the information that they actually pass on. Know them, understand them. All right, cranial and spinal nerves. So we have 12 pairs of cranial nerves, that connect to the brain and the brain stem. Okay. 4 classifications. So they can be sensory nerves. Here's the type of information that these sensory nerves carry. Special sensory nerves, carried by highly specialized neurons. Okay? You can't smell with your touch receptors, or you can't see with your pressure receptors. Okay? So these are highly specialized. Your motor nerves and your mixed nerves. Okay? So 4 groups. They are generally identified using Roman numerals. 1 through 12. Very specific names associated with these with these cranial nerves. I've stuck in a mnemonic there. Google has much more colorful mnemonics, which I'll probably get fired if I stick up here. So do look at Google and try to remember these names and their functions. Very important as well. Okay? As you'll see, some have pure sensory functions. Some have pure motor functions, and others have both. Okay? Here are the cranial nerves themselves, obviously not showing you the nuclei, which are located deeper in these structures. Okay? Locations of these different cranial nerves. Number one, notice, is olfactory, so you don't actually see it here because they're olfactory tubercles. They actually run on the inferior aspect of the brain. Okay, so you can't see that in these diagrams. There's the optic nerve, the optic chiasm, and then the optic tract. The origins, as I mentioned before, and their functions. Okay? Has anyone printed these notes? Oh, tell me what tell me where the lecture stops. Tell me. Oh, you printed. Good. Yeah, yeah. Do you have any sense? Thanks. Sorry? 129. 129. We're at 6. Okay. Yeah. Keep going? Okay. All right. We'll see you next week. Guys, I had only one question. I was very impressed. Somebody studying during the break. Okay, I have only one question so far from this class. Please ask the questions early. Don't wait till the last minute. Remember, my email stops working week 13. Oh, I still have 3 minutes.